Hello my dear friends, let us discuss today about the post-fertilization structure and events. Among that, firstly let us discuss in this session especially about the post-fertilization structure, that is the endosperm. As we all know how endosperm is developed during the process of double fertilization. Let me just remind you, in case of double fertilization, the first male gamete will fuse with the egg or the ovum to form the zygote. So zygote after fertilization, so it will undergo mitotic division or the cleavage to form the embryo. Whereas the primary endosperm nucleus is formed by the fusion of the second male gamete with the secondary nucleus or the polar nuclei. So this primary endosperm nucleus in fact will undergo the mitotic divisions to form what we call it as the endosperm, which is in fact the nutritive tissue. It is very important as the nutritive tissue for the developing embryo. This provides the nutrition for the embryo. That we need to remember, my dear friends. Generally, friends, that endosperm is triploid in nature because already we know that two haploid nuclei fuse to form the secondary nucleus inside the embryo sac. So later the male gamete nuclei also fuses with it. So three haploid nuclei fuse to form the triploid nucleus. That's why we call the in fact fusion of the second male gamete with the secondary nucleus as triple fusion. So during the process of triple fusion, there is formation of endosperm. Not only 3N, my dear friends, it can also be diploid, that is 2N. Let us say for example we have lily. So in case of lily it is diploid. The same, it can also be polyploid in many different plants, even up till 15N it can be. So other than this, let me tell you that the endosperm especially has certain important hormones called cytokinins. These cytokinins are very necessary for cell differentiation. So not only this, my dear friends, the outer layer of the endosperm has allurone layer, that is the protein layer, which secretes an enzyme called amylase. So this amylase enzyme is necessary for breaking down the complex sugars into the simpler ones. Generally, in the case of endosperm, we find that the starch is highly stored as the reserve food material, but in few cases, the starch can be replaced by oil, as in the case of castor. So these are the points we need to remember. Okay, for example, white flour, which we use for example bread making, my dear friends. So that will have what endosperm. In case of dicotyledons, the endosperms will be in fact replaced by fleshy cotyledons, so which we consume in fact. So remember that in case of monocotyledons, generally endosperm will act as the important nutritive tissue. So as I said you earlier, there are two types of endosperm. Alright, one is seeds, endospermic seeds, and non-endospermic seeds. That is with respect to seeds we can say. So endospermic seeds are also referred to as the albuminous seeds, in fact, where a part of the endosperm will be left over. Generally in case of monocots we can find, as in the case of for example wheat and maize, as well as in the case of paddy we can find endospermic seeds. Secondly, non-endospermic seeds are ex-albuminous seeds, where endosperm is completely utilized by the developing embryo. So there is no endosperm which is left over, as in the case of pea and beans we can find. So this is a short introduction about the endosperm. So endosperm is a post-fertilization structure. It's a nutritive tissue which is necessary for the developing embryo. Dear friends, next let us discuss about the types of endosperm. Just now we discussed how endosperm acts as the nutritive tissue for the development of embryo. Here we have three types. One we have here is the nuclear endosperm, then we have cellular endosperm development, followed by the HELOBIAL endosperm development. So here, my dear friends, let me tell you that the nuclear endosperm is a type of endosperm where we can find much of karyokinesis, which leads to the formation of thousands of nuclei form, which are generally pushed towards the periphery, towards the corners. So after the karyokinesis, that is after the formation of nuclei, thousands of nuclei, there is no formation of the cytoplasm. So karyokinesis is not followed by cytokinesis generally with respect to the nuclear endosperm. This point you remember, my dear friends. And let me tell you that further how the formation of the cell wall occurs. Generally, from the periphery towards the center, there is formation of the cell wall. So this you need to remember, my dear friends. First, there is formation of free endosperm nuclei, and later there is development of the cell wall, that is from the periphery towards the center. So meanwhile, what happens is the zygote, which was one cell, it keeps developing into the embryo by utilizing the nutrition which is present in the endosperm. As I said you, generally starch will be the reserve food material with respect to the endosperm. So starch will break down into simple sugars with the help of the amylase enzyme, which is secreted from the outer layer of the endosperm called the alluron layer. So when it breaks down, it will utilize anyway. So not only that, even for, as I said you, even the seed it provides the nutrition. Furthermore, my dear friends, this nuclear endosperm, the best example we can bring out is the coconut. So where a large central vacuole will be left over at the center, which forms a liquid endosperm in case of coconut. So that is, coconut milk will be stored here. So whereas the coconut flesh will be formed after there is a formation of the cell walls. Like that, we can find with respect to the coconut. Not only with respect to coconut, my dear friends, the nuclear endosperm can also be seen with respect to, we have maize, we also have wheat, cotton, and to go with, as I said you, coconut we can take into consideration. Remember that endosperm is an important nutritive tissue with respect to monocotyledonous plants, but not generally with respect to dicotyledons. In case of dicotyledons, as I said you, an important point, my dear friends, remember, there is the fleshy cotyledon which replaces the endosperm generally. So we have second type of endosperm, which is the cellular endosperm. Here, remember that the karyokinesis, that is after the division of the nucleus, immediately there is a what I can say cytokinesis which occurs. Immediately it divides the it forms the cell. Karyokinesis immediately followed by the cytokinesis. So when the first division occurs, generally that is the transverse division, there is a formation of the two chambers. The larger chamber is, we can refer that to as the micropylar chamber, and then we have the smaller chamber called as the chelazal chamber. So in fact, further, it keeps dividing to form the cellular endosperm. As I said you, meanwhile, the zygote keeps developing into embryo. So this type of cellular endosperm formation can be seen with respect to petunia. It can also be seen with respect to datura and also with respect to balsam. So there are so many other important plants we need to remember. Then, my dear friends, we have next the final type of endosperm, that is the HELOBIAL type of endosperm. So we refer this HELOBIAL type of endosperm as intermediate type. Note this point, because it shows both the nuclear type of division as well as the cellular type of endosperm division. So in case of nuclear type of division, there is the formation of peripheral nuclei which can be formed with respect to the HELOBIAL endosperm. But generally there is first formation of micropylar chamber and the chelazal chamber. So there is a division. Later there is the development of these nuclei into further the nuclei will form the cell wall and develop into the endosperm. right? So as in the case of nuclear endosperm we observe here, we can find. So then the other nuclear endosperm also, in fact, we can how we found the division, that also we can find, children. So anyway, so both types we can find with respect to helobial type of endosperm. Here also there is zygote, further the zygote will develop into embryo. So anyway, so these are the three different types of the endosperms which we need to take into consideration. And also remember that there are three families of angiosperm which does not produce endosperm at all. So let me tell you that. First we have Podostemaceae. We also have Orchidaceae, and we also have Trapezae. These three families generally does not produce the endosperm at all. Other than these three families, most of the other families will definitely produce the endosperm. So keep watching the channel BIOZEE classes and keep updated with the upcoming videos. And I request everyone to please like, subscribe, share, and also give your comments, valuable comments in the comment section, so that definitely there is so many important aspects which can be dealt further. If you are interested in many topics, definitely you can just put back in the comment section. Definitely I shall deal about that. And follow the channel with much interest, I request everyone. So thank you very much.